And probably the best study of these is protein glycation. When you have an excess of glucose in your blood, you get non-enzymatic modification of proteins by glycosylation. And that can be useful. You can measure the hemoglobin A1C and use that to follow diabetic patients. It can be potentially deleterious because it can influence protein function. But you're only going to see this on proteins that have a long half-life. So cells that turn over rapidly, I have a hard time understanding how they may be impacted by diabetes. Alveolar macrophages, though, are very, very interesting cells. So the resident alveolar macrophages in the lung, in a mouse, are a non-dividing population whose lifespan is equal to the lifespan of the mouse. We can tag these cells with a GFP-expressing lentivirus, and we will see the same population of cells present in that mouse 9 months later. So these cells are hanging around long enough to be impacted by diabetes. That is the direction we'll explore. Just very briefly then, just to quickly sort of finish up, we have looked a little bit at T cells, and the T cell story is kind of interesting. What we found in vitro is that, or ex vivo I should say, if we take T cells out of a chronically diabetic mouse and we stimulate them with plate-bound anti-CD3, we see that the diabetic T cells proliferate more than the non-diabetic T cells. And we see that whether we use splenocytes or whether we use very purified T cells. And again, the effect is not enormous, but it's a very, very reproducible trend with at least statistical significance. And I think that it's largely a T cell intrinsic effect because in this experiment, this is in the absence of antigen-presenting cells. We also see, and Therese likes proliferation. I think actually cytokine expression sometimes is more sensitive for looking at T cell activation. And in fact, we see increased expression of interferon gamma, IL-2, TNF, GM-CSF, IL-12, IL-17, as well as IL-10 and IL-13, all more produced from the diabetic T cells after anti-CD3 stimulation. No difference in IL-23. So to look at this in the context not of an antibody binding to the T cell antigen receptor, but actually an antigen hitting the T cell antigen receptor, we used OT2 transgenic mice. These mice have a transgenic T cell antigen receptor that recognizes an ovalbumin peptide. And we find that we can see increased proliferation again in the diabetic mouse. So this suggests that it's not just an artifact of anti-CD3, of the diabetic, of the T cells taken from diabetic mice stimulated with this antigen. We've also done this in vivo. And in vivo, it gets really complicated. So we have evidence in vitro that the diabetic T cells have kind of a hyperproliferative state. So we've looked at this in vivo. We did splenocytes coming from healthy OT2 mice. We labeled them and adoptively transferred them into either diabetic or non-diabetic mice. We then stimulated these mice with ovalbumin protein, and we presented this to the lungs. We used a technology that I won't go into of packaging antigen in a glucan particle that can go down the lung. It's ingested by dendritic cells through the dectin receptor. The dendritic cells go to the thoracic lymph nodes and prime the adoptively transferred OT2 cells. And here we see that the suggestion maybe of sort of two things that are very interesting. At 72 hours, we put the control T cells into, we put the T cells into a non-diabetic mouse, they proliferate more than when they go into the diabetic mouse. So this is the opposite of what we saw in vitro. In vitro, there was this hyperproliferative state of T cells, but if you put T cells into the diabetic environment, they seem to be suppressed in proliferation. And the last experiment I'll show is this sort of crisscross experiment where we now made the OT2 mice diabetic or not diabetic, and we transferred their T cells either into a non-diabetic host or a diabetic host. And again, we stimulated with the ovalbumin delivered to the mucosa in the airways. We took the thoracic lymph nodes out 3 days later. And we see in the non-diabetic host that you have actually the diabetic T cells are now hyperproliferative in the non-diabetic host. But in the diabetic host, both the diabetic T cells and the host T cells have reduced proliferation. So this is an evolving story. All I can say at that point is it's sort of interesting, but I'm not quite sure what the implications are. It seems that there is an intrinsic effect on T cells for hyperproliferation, but some environmental effect for suppressing T cell growth. And then just the biochemical mechanisms, again, all of the complications of diabetes through these different pathways are driven by hyperglycemia, which is sort of our motivation for using the simplest model, the streptozotocin. And the very last thing is just to link to the humans and then we'll be done. So from the mouse data that we have so far, this is an evolving story, but from the data we have so far, we can make some predictions about what we would expect to see in people who have diabetes and TB. So the mouse data, again, is a delayed response to infection and late control of replication, but then later an exuberant immune response. So the predictions are that, first of all, TB risk will correlate with the severity of diabetes. We have to make mice diabetic for 3 months, or at least 2 months, to see the susceptibility. Second is that we might see higher bacterial load, both in the diabetic people who have latent disease or in those who have active TB disease. Third, we might expect to see more post-primary TB in diabetics. That is, people who become infected with TB and never develop latent infection. They go straight to disease. And the way that we identify those patients clinically is that TB disease occurs in the lower lung zones. Reactivation of TB is typically in the upper lobes. Post-primary TB is typically in the lower lobes. And in fact, there is clinical evidence that there is more lower lung zone TB in diabetics. We might expect to see worse lung injury and higher cytokine levels in the diabetic hosts, and perhaps as a response of higher bacterial loads and maybe more inflammation, perhaps a poor response to treatment. And there again, there is some evidence that diabetic patients with TB clear the infection more slowly than non-diabetics. So the last 2 slides is one little bit of evidence. This is from a friend of mine, Blanca Restrepo, who follows a population with high rates of TB and high rates of diabetes. These are Hispanic people living in northern Mexico or southern Texas on the border. And in that population, she took people who have diabetes, who had, or not diabetes, but all who had tuberculosis. She took their peripheral blood and she stimulated with antigens. So she's essentially doing the quantiferon test, the interferon gamma release assay, but she measures for additional cytokines. And what she found was that for TNF, IL-2, interferon gamma, and several other cytokines I didn't list here, the diabetic patients with TB make more than the non-diabetics. And the higher the hemoglobin A1C, the higher their cytokine levels. This, again, to me was comforting because this is similar to what we see in our diabetic mice with chronic infection. They have more cytokines, higher cytokine generation. And the last little bit, we're doing some clinical studies in my group, collaborating with a hospital in South Korea that's a national tuberculosis hospital, 300 beds, and we're able to take advantage of an existing study and pull data from a database from a study that has about 600 recruits who have either new diagnosis of TB or relapsed TB. Looking at a number of correlates in the diabetic population, we determined that about 25% of their TB patients have diabetes. In that population, we looked at a number of parameters. I'm only going to show one, which is death. And if you have diabetes and TB, you have about 3.3-fold greater chance of dying. And interestingly and disturbingly to me, that risk is greatest in people who are under the age of 50. So diabetes appears to promote worse TB outcomes, very substantially worse TB outcomes, and is doing that in a relatively young population. So this really does, I think, is a topic that kind of merits better understanding. almost all of these deaths are TB-related deaths. The people with diabetes weren't dying from heart attacks or strokes. They're dying from hemoptysis and from respiratory failure. So I'm going to stop there. I think I went over. I just want to merci de votre attention. Almost all the work I showed you was done by Therese Fowler-Scott, a postdoc in my lab, also some by Greg Martins. My technician has helped with this, and my wife has helped with the clinical study. And we collaborate with George Reed on the clinical studies, epidemiologist. Chris Sassetti, microbiologist, has helped us with some of this work. The work in Korea is a collaboration with Laura Vai and Cliff Barry from the NIH and the investigators in Korea. And we're supported by funds from the NIH as well as some pilot money from the university. So I'll stop there. And if this talk goes on forever, but I'll stop and I'll let you out. We have a second piece about cholesterol and lipids and host defense, but there's no time for that. But I'm happy to talk about that later. So thanks very much. Arnold, please.
I have plenty.
I have one comment. By listening to your talk, it was very, very interesting to see. At the beginning of your talk, you defined gamma interferon as the real susceptibility factor. But I think this point of view now can be criticized because by in vivo investigation, it is clear that the prediction of the first cartoon that in patients with tuberculosis and diabetes, we should have less gamma interferon than in other patients. In fact, it's wrong. It's wrong. It's true in your experiments in mice. In our experiments with other infectious diseases, it's also wrong because we found that in fact there is an activation in the patient. So the main question probably is the defect of regulation better than the defect of critical. It's very interesting because if you look at gamma interferon, we do not find anything different. Ions, we do not find difference. But if you remain, or if you come back to the initial paper, everything says the main factor for tuberculosis was as an ions. So everything is wrong. So perhaps it's time to rewrite the immunopathology of some infectious disease.
Yeah, I think I agree with you. And I think that it is a fault or a problem of kind of a simplistic ideas about susceptibility and resistance, virulence and attenuation to sort of make things black and white. And I think it's as we said earlier, in tuberculosis, the disease is from the immune response. And it's really that regulation of the immune response. And one thing that we haven't looked at yet, but ultimately hope to look at, is whether there are deficits of counter-regulation in the diabetic host. It could be that they make this protective immune response, but then for some reason fail to shut it off, in which case it progresses from being a protective response to being, a destructive response. And our diabetic patients who are dying at a 3 times greater rate than the non-diabetic patients, what they're dying from is lung injury mediated by their immune system.
Just one question. I want to know if you have made some study about the anti-inflammatory response.
Although I work in an inflammasome-rich environment with the Goldenbach and Fitzgerald labs next door, so far we have not looked at inflammasome. We do see that the diabetic mice are producing IL-1 beta at levels equal to, and in most cases exceeding, the non-diabetic mice. There is, so I think that the inflammasome per se, I don't think that there is a defect or a fault. Maybe there is some excessive activation or failure to regulate the inflammasome. And there actually is an interesting story that's evolving from the laboratory of Christopher Sassetti, my colleague, the other person at UMass who works on TB, where they're finding actually that interleukin-1 regulates the inflammasome. That interferon gamma regulates the inflammasome. And that macrophages that are pre-treated with interferon gamma have reduced processing of pro-IL-1 beta to IL-1 beta.